Our lab is specialized in whole genome sequencing analysis, and we decided to investigate the mechanism of resistance to CAR-T19 therapy in large B-cell lymphoma in collaboration with the Moffitt Cancer Center and the University of Miami. And despite the cohort was not large, but the quality was extremely high because we were sure about the purity of the samples. We were sure, thanks to whole genome sequencing, about all the genomic features are essential or critical or driver for each cancer type. And what we found is that there are distinct genomic features like APOBEC mutational activity, like certain deletion, like certain present complex mechanism like these double minutes where an oncogene is amplified multiple times, like 7, 10 copies of MYC, for example. And all these mechanism tends to be observed only in patients that relapse. So if you combine all these different genomic features, you can see that the patients with these genomic features, they all relapse after CAR-T, and the patients without, they almost never relapse after CAR-T. And because CAR-T is like a historical groundbreaking drug that is going to change, that is changing our practice, in particular after this ASH and after the New England paper from Fred Locke, where probably is going to be approved in second line for diffuse B-cell lymphoma. Understand why 50% of patients fail CAR-T is essential, and I think genomics can help, but also we show with others that the CAR-T killing activity is not just the CAR that bind the tumor cells, but there are probably other elements that other people also support like with their data, this hypothesis, that CAR-T is kind of like a booster and promote the immune system to, wake up again and attack the tumor. And that's, I think, is another important point that people are already investigating. We're not the first and we're not the last, and a lot of people are working on this, and hopefully a better understanding of CAR-T and better understanding of the interplay between immunotherapy, immune system, and cancer cells will really allow to develop a more precise therapy, adapt therapy to the patients and to their tumor biology.